Another day. Welcome everybody. Good to see you all again. I have a lecture today on metal complexes as enzyme inhibitors to start with. If you think back for a second the things we discussed yesterday and the day before, I mean, we had this discussion actually yesterday already, when we discovered that many of the metal-based drugs that are, for example, in use as in the platinum drugs, also when we talked about the ruthenium drugs, the ruthenium-based ones, we talked about the titanosines, all these compounds we discovered have a similar way of activation, if you like. Yeah? We have the hydrolysis of a metal halide bond or a metal carboxylate bond, or let's say just a metal X bond that is hydrolyzed, and then we arrive at equated species. So like, for example, the platinum complex that has a water complex bound, and then this complex actually binds to DNA and then has some function down the line. So all these compounds were prodrugs, as we said, and also all these compounds had, in a way, a similar mechanism of activation by being hydrolyzed first and then forming an aqueous complex, and that aqueous complex then has some follow-up action. It could actually, as we saw yesterday, it could actually react with proteins, enzymes, or with DNA, or with something else. So a lot of these things had in common this mechanism of being activated, and we discussed already yesterday, before one of the lectures, I think we already discussed that maybe this is actually something that is special for metal complexes, and it's a feature, it's a particular feature of metal complexes that can be used beneficially. That is relatively uncommon in purely organic drugs, so it gives us another mode of action, another mode of activation beyond purely organic drugs. This lecture today will serve to demonstrate that this is not always the case. Now, we can have inorganic compounds that serve as drugs that are not being hydrolyzed, that don't lose any ligands, that are just very, very stable, and they act just as they are. There is no transformation taking place, but they really act as they are, and they don't need this. So it adds to this overall picture of what metal complexes can do. They can be hydrolyzed, special reactivity. They can be redox activated, electron out in. They can be photoactivated. Here is something where the metal complex does not need to be activated or reacted or transformed in some way. But in the cases that I will discuss today, at least I think in all of the cases, at least in most of the cases, you will see that the metal complex actually remains intact and has a function while being intact. Okay, so that is the overall scheme, and the compounds that you will see, mostly they will still be concerned with anti-cancer therapy, and then we'll move on to other indications later today in the second lecture. So let's look at some of these things, and the one example that maybe has received, I was going to say has received most attention, but maybe that's not true, but an example that has received a lot of attention is the case of inhibitors for protein kinases. And I will start out by explaining what protein kinases are, and then we come to the inorganic aspects and what people have contributed in this field. So protein kinases are very, very common class of proteins, of enzymes in your body, in my body, in mice, in chimpanzees, in hamster, whatever you want. They're everywhere. Protein kinases are everywhere. What do? They modify enzymes by phosphorylation. And that very often is a signal then that something should either be started or something should be stopped. So very often an enzyme has whatever function it has, but it is really activated only when the when the side chains are phosphorylated. So it can either be serine or threonine or tyrosine, and those are phosphorylated, and then they bind differently to their target, and then something stops. Very often this has to do with cell signaling. So it's not an enzyme switching on or off, but it's a signaling molecule that will then go up. You can see here it is estimated that the human genome has about 500 different protein kinases. So that is really a large number. This is about 500. It's about 2% of all human genes are contained in those kinase-related genes. You can already see it's a very, very big group of compounds. I will talk about some screens and some assays, and those contain about 300 or 350 or so kinases, but it's estimated that there are many more that we actually don't know yet. Yeah? So it's a really large number, and they regulate, as I said, those kinases, they regulate something. Something goes either up or it goes down, and they regulate basically all aspects of cellular life. Yeah? So you see, it is estimated that 1/3 of all human proteins are modified by kinase activity. Not maybe directly, yeah, but through something else that is phosphorylated, and then that influences this enzyme, and so on and so on. So, you can see this is ubiquitous in your body, in my body, in, as I said, chimpanzees and mice as well. And it's very, very, very important. So if you interfere with it, you potentially have, for example, the possibilities to stop cancer growing very easily, if you just find the right one. But of course, you also have the problem that there are 499 similar kinases, and of course, you don't want to affect those. You want to affect only the one that is related, for example, to cancer growth. So we will come to this point of selectivity. We will come to that in a moment. We've been talking about diseases, and here you can see kinases are important therapeutic target. As I said, they up or down regulate something. For example, they upregulate apoptosis in cancer cells. So if I can upregulate apoptosis, it means that this cell is going to die more readily. Yeah? And if that's a cancer cell, this would be a way of killing a cancer cell very easily, very straightforward, at least conceptually. Yeah? And for that reason, those kinases are really important therapeutic targets, not just in cancer, but in almost any disease. Yeah? You can imagine there are so many different things that you, by slight regulation, you can improve, be it your heartbeat or your blood pressure or diabetes related or whatever, anything, you name it. There is certainly a kinase that regulates something in that path. And if you can target, if you can identify that kinase and you can target it and say either up or down regulate, whatever is required, you can certainly improve that condition. It's just a question of finding the right one and then addressing it. One compound that has turned out to be one of the most potent lead structures is this compound here. It's called staurosporin. It's a natural product. It has been isolated already in the 1977s. Interestingly, it was isolated from a bacterium, Streptomyces, and this compound was shown to inhibit or interact with many, many, many kinases. How does it do that? There is a cartoon-type picture. There are also crystal structures available, but I think the cartoon-type picture is probably easier. All of the kinases that we know, they require energy. So in order to transfer the phosphate, we need a phosphate in the form of ATP. So this is adenosine triphosphate. And one of these phosphate groups is actually transferred. So there's always an ATP binding site here. And then this phosphate is transferred to something else, and then that something else is up or down regulated. So one way of modifying the activity or inhibiting, in fact, these kinases would be to put something in this pocket. Here is the adenine, and then there is the sugar, and then the 3 phosphate groups. But if you put something in this pocket here, and so the adenine cannot bind, then you can't have phosphatase activity, or kinase activity rather, because the adenine doesn't bind anymore. The substrate doesn't bind anymore. And staurosporin, this is the molecule in a slightly different view, does exactly this. It is made, as you can see, it has these very nice hydrogen bonding capacity here, hydrogen bond acceptor, hydrogen bond donor. Staurosporine is much the same, hydrogen bond donor, hydrogen bond acceptor, and some bulky group to the left, to the right, and to the middle here. And that fills out this pocket very nicely. ATP cannot bind anymore. Phosphate cannot be transferred. So clearly, this is a clever way of inhibiting kinases by simply blocking the ATP binding site. I should say that potentially there are other ways of inhibiting kinases. You can inhibit at other sites. You can, for example, block this transfer of the phosphate here. You could do that. But I should say that really targeting the ATP binding site is the most common way of inhibiting protein kinases. Yeah? There are others. If you look in the literature, you might find. But this is, let's say, the most common. And this molecule, staurosporine, as I said, it was isolated from this bacterium, Streptomyces. It has a relatively complicated structure. If you look at it, this is the structure. This is not so difficult, but this part here, you know, the stereochemistry has to be right, and then you have all these substituents here. So the total synthesis of staurosporine actually takes a couple of steps, and it's not really straightforward. So when you just look at the structure, this is where the metal chemistry comes in. I mean, this is a flat molecule. That's not so difficult to make. But this three-dimensional part here is actually quite challenging, you know, to get all the stereochemistry right. And this is where my colleague Eric Megas comes in. And Eric had the idea, why can't we model this part of the molecule here by putting in a large inorganic fragment. And then we can use this part here and leave it as it is. There are two nitrogens here. Those are very good ligands for a metal. And this is the general structure they came up with. So you can have this sort of, well, let's say it looks a little bit like a phenanthroline type structures. It's a little bit larger. It has, in the opposite end here, it has hydrogen donor, hydrogen acceptor capacity. And then instead of this complicated organic fragment with the various stereocenters, they put in a metal complex with 4 ligands. If this is an octahedral complex, you very quickly make up this big spherical arrangement, yeah? Much like this is here, big and spherical and three-dimensional. So this here basically has the same function. And you can look at this and say, well, here is the hydrogen bonding, the pocket filling part, and this is this big sort of mimicking this rest here. So that's what they did. This was their rational design idea. And they started out by working with ruthenium-2 complexes in an octahedral arrangement, partly for the reasons that we discussed previously already. Ruthenium-2 is relatively stable. It's not very toxic. It's substitutionally relatively inert. Yeah? If you have ligands on it, they will stick to it most of the time. So ruthenium-2 seemed to have some good functions for this kind of purpose here. And then they did some modeling in order to confirm that their idea was actually also valid. And if you have something like this, you make a molecule like this with maybe some substituents here and some new rings. This looks pretty similar already. It turns out that this really binds very well, and this three-dimensional ruthenium here actually serves to fill the upper part of this pocket quite nicely. So the whole design idea, substituting this part by a bulky three-dimensional ruthenium, actually has worked really, really, really well. And in this way, you can build fairly complicated structures, yeah, relatively easily by simply changing those 4 substituents on the ruthenium. So there's X, Y, Z, and W, X, Y, and Z. Those 4 substituents, if they can be different ones, and you can have very large ones, or you can have like, you know, maybe a chloride or a carbonyl, just sort of linear, small ones. So you have a whole variety of things you can change here. And if you think of it, and we mentioned already 500 different kinases, you can imagine if you play around with these 4 substituents, and they can be made larger or smaller, or maybe sort of more looking in this direction or more in stretching that direction, you can already imagine how you can make these compounds here very specific for certain kinases. So it's more a question of synthesis, really, how do we make these compounds, than anything else. Okay. We already mentioned why ruthenium. Here is summarized. We can quickly go through this. structural variety I just explained. Here is again our 30 stereoisomers, which reminds me that I actually forgot to look this up and maybe draw them. I think if you actually, I think there must be at least one of Eric Mega's articles. You know, in his lectures he has a slide where you can see all the 30 different isomers. So I would think he has put it in one of his review articles. So maybe we should have a look. Anyway, great structural variety. A, B, C, and D can all be changed. So this is 4 different substituents in just 4 different ones and in different spatial arrangements. So there's quite a lot of variety here. Air and water stable. General toxicity isn't very high. So they look like good starting candidates. And let's go ahead with this. Let's look at this. Here is some examples from this research program. Those are different inhibitors that have actually been used, and nano or even picomolar affinity has been achieved. So instead of ATP, these compounds bind, and you see here this IC50 number is an expression of affinity. The lower, the better. This is the concentration that inhibits 50% of the enzyme or 50% of the activity. You can see here there's one that is 130 nanomolar, which most people would already consider, pretty good. Less than micromolar, 0.1 micromolar, that would already be pretty good. But then look at these numbers here. This is even, what is like 1, 2, 3, even 3 orders of magnitude lower, almost. So this is spectacular affinity. This is 400 picomolar affinity. So that's really, really, really very, very, very high affinity. Absolutely fantastic compounds. And you look at this molecule, I mean, okay, you have this heteroaromatic part here. I guess there is some synthesis involved in this. It's maybe a few organic synthesis steps. But then there is the CP ruthenium carbonyl fragment, which is really easy to make. I mean, the inorganic chemistry that goes into this fragment is relatively simple, really. This one as well. You have a ruthenium, carbonyl ligand, chloride ligands, very standard. And then here you have a Schiff base, a pyridine Schiff base derived type of ligands. Okay, there's a phenyl group here, but also to make this is maybe 2 or 3 synthesis steps at most, and then comes in the ligand. And here is one that has the cyclic ligand with the sulfurs and the carbonyl, and that one also has spectacularly low affinity. These numbers here, EA1, FL172, PIM1, those are different kinases. Those are abbreviations for certain kinases, and you see that those 450 picomolar affinity is specific for this one particular kinase called PIM1. Okay, so those are, super specific enzymes for these particular kinases. Now, if you step back for a second and look at these structures here, as I told you, Megas and his group started out by doing ruthenium chemistry for the reasons we outlined. Also because ruthenium chemistry is relatively well established. There are many, many starting materials that you can use with, either cyclopentadienyl ring or Schiff-base derived ligands or diimine ligands or, chelating ligands like this one here. This is the sulfur triisocyclononane, but there's also other ones. So many of those starting materials are already described in literature, which makes it very easy to initiate such a research project. You can have a really well-defined set of starting materials already, and then all you need to do is some organic chemistry here at this end of the molecule. But, of course, the design principle, I want to build an octahedral complex which binds these 2 nitrogens, and then there are 4 other coordination sites that I can occupy. You can transfer this to many other metal complexes. For example, we had already in the lecture on Tuesday, we had octahedral platinum-4 complexes. And you could also see that a great variety of those can be synthesized. You can have osmium complexes. Osmium is right below ruthenium, so it should in principle do similar kind of chemistry. You can do iridium chemistry, which works very, very nicely, actually. There is a lot of iridium chemistry behind it here that leads you to octahedral complexes. I think I will show you actually some synthesis examples on octahedral complexes. Osmium is an interesting one because osmium can very easily be used. The main trouble in osmium chemistry is that there are less good starting materials. So osmium chemistry is by far not as well developed as ruthenium chemistry. And if you want to do such a project and you want to put on, all kinds of different ligands, you will quickly see that osmium is much more limited, not because it has a different chemistry or it's, for whatever chemical reasons, but there aren't simply as many suitable starting materials as there are ruthenium starting materials. So that's a main limitation in osmium chemistry as opposed to ruthenium. By the way, many people think that osmium is horribly toxic, and you shouldn't touch osmium. I guess this is because of osmium tetroxide, which you all know. It's a well-known oxidation reagent, organic chemistry. You do this epoxidation reaction with osmium tetroxide, alkenes, and osmium tetroxide. Yes, of course, osmium tetroxide is very toxic. It's a volatile compound. Yeah, it attacks anything that it sees, adds to double bonds, and so on and so on. Yeah, osmium tetroxide is certainly very toxic. But that doesn't mean that all osmium compounds are toxic, because osmium tetroxide is, of course, very special also. It is high-valent oxidant, osmium. It's oxidant in the oxidation state +8, if you believe it. Yeah, I mean, it has 4 oxygen ligands, and that's all it has. This here is formerly ruthenium-2. Yeah, 1, 2, yeah. And also the other ones here. They're formerly in the oxidation state +2. So it's a totally far apart oxidation state to start with. Yeah? And then osmium tetroxide, of course, has this particular reactivity because of the osmium-oxygen double bond that attacks other double bond, and so on and so on. So osmium tetroxide really is a very special compound. And yes, osmium tetroxide is very toxic. You need to be super careful if you work with it. But that doesn't mean that any osmium compound in the world is very toxic. So again, this is a perception that people might have, but you are, really well-trained chemists. Many of you are inorganic, and you can certainly tell the difference between osmium tetroxide, osmium oxidation state +8, and an osmium compound of this type, where it's octahedral and +2. And in fact, osmium is very inert. If you think of the speed of ligand exchange reactions on osmium, let's say +2, it's actually very, very slow. So in a sense, these compounds, if there's osmium here, are very, very inert in terms of ligand exchange, which is what you want. You want these to be very, very stable, as we said. Okay. So again, it just comes to the question, which way do you get a certain compound? Metals aren't always necessarily toxic. It depends how you prepare them and how you wrap them up in organic ligands. Okay. If you look at these molecules here, let's just take those 3 which are up here. You see there are subtle changes. There are some things which are similar. It's always this part in the backbone because of the hydrogen bonding we can understand. But then, for example, here's a bromide, and here's no bromide. And here's a fluoride, and that isn't on all the other compounds. So there are subtle changes, and they make for the selectivity. And Eric Megas and his partners, his group in particular, they have been initiating a project where they also do a lot of structural work, and I think I stopped counting at a certain stage. They have certainly more than 24 crystal structures of the various different kinases with a target bound to it. This is, for example, one of these compounds. This is FL172. So FL172 is the one in the middle here. This particular ruthenium compound was inserted into this kinase, and then they got a crystal structure. And here you can see very nicely, this is the ruthenium here. This is this sort of imine-derived ligand. Here you have the hydrogen bonding donor and acceptor in the backbone of the molecule, and you can nicely see how this pocket here in this particular kinase, this sort of the flat ligand inserts into the pocket to the back. But you can see that up here, there seems to be an open space. And this is really nicely filled by this pyridine ring. Maybe in another kinase this does not exist. It's more like this. Then, of course, this molecule wouldn't fit anymore, and you would lose a lot of the affinity. And here again, same thing up here. There seems to be some kind of of space, and this phenyl ring very nicely slides in and fills this space here. So this is a very nice design. I guess when you see it, you see how well it fits. You say, oh, well, cool, how clever. Yeah? But you really need the crystal structure of the enzyme first, and then you can think and say, okay, now let's see, what do we need? Do we need another phenyl ring here? Maybe there is something, let's say if there would be a little more open space here, maybe you want to attach a chloride or a bromide atom in this meta position here, if it's here. And if the space is up here, maybe actually a chloride would fit in here quite nicely. Yeah? So if you have the crystal structure of the enzyme, then you can design your ligand. And, as I said, I don't know how many, I can't guess, 100 compounds certainly have been made, maybe more. And they all are very specific for certain kinases. Here's another crystal structure of another compound. Here's a CP ring, cyclopentadienyl ring. So there seems to be some space as we come into the molecule. There seems to be some space here, and the rest isn't really that much. So you can see really there is a very high selectivity and very specific binding. Talking about selectivity, this is just one of the many, many, many examples that are in the literature now from Eric Megger's group. You can see this is a publication in Angewandte Chemie in 2010, so it's already 8 years ago. Just for a change, I have chosen an iridium compound here. This paper is about iridium complexes. I've chosen an iridium compound. It's also iridium-free, as you can see. 2 chlorides, 1 negative charge here, so it's iridium-free. If you think iridium-free really has very, very slow ligand exchange, so this again is kinetically a very stable compound. It is octahedral. It has chloride ligand. It has this, our hallmark ligand here with the hydrogen bonding capabilities. And this compound was tested against a panel of 229 different kinases. So it's about half of the total. Yeah, 500 total estimated, maybe even a few more, and 230 or so, 229 to be precise, were investigated in this compound. And this is the number of protein kinases, and this is the remaining activity. So 100% activity would be, up here. So 182 kinases were not affected. They had full activity in the presence of this inhibitor, meaning the inhibitor doesn't quite bind to this kinase. But there are some here, 5 kinases out of 229, that had less than 50% activity. So this compound is quite specific here, only those 5 here we really need to worry about. And then we can start working on it. We can try and get crystal structures of all 5. Maybe some crystal structures are known. We can do docking experiments or co-crystallization experiments, and then we can see how and why this compounds here fit so well into those few pockets here. And then we can maybe make it even more specific. If we see, oh, there is one here that's interesting, but it has some more space here, maybe we add another iodine atom, or we add 2 methyl groups to this side, or whatever, And then we can make it more specific. And this is the way that we get this very high specificity for only a small number of kinases. And as I said, this is just one example. Typically in Megger's group now, when they have a new group of compounds, either they come from a known crystal structure and say we want to build something that fits this pocket, or they come from a new compound class and do a screen against, I think their number is now up typically to 300 or so kinases. This is a profiler assay. So you actually buy, in this case from Millipore, but I think there are others now, you buy from a company these 96 well-played based assays where you have all the kinases in it and some clever assay to monitor activity, and then you can do in a plate reader, much like you have in your nice facility over there, you can do a binding assay, and you have some colorimetric readout that tells you whether your compound inhibits the kinase or not. And if you have 300 in parallel, you can imagine you get very detailed information. And some may be really good inhibitors, and a large number may not be affected very much. Okay, so that is a really big program that Megger's group has started about 10 years ago or so, and now they're moving, I think, some of their best compounds ahead. Of course, they patented compounds when they find a really from one of the biotech companies. It's not Bachem, but one of these sort of suppliers that are very specific kinases for a certain important kinase. So people use it actually already in biochemical assays. They provide certain specific kinases that have some relevance, and you can study, for example, what happens in your cell culture if you inhibit this one kinase very specifically. These compounds, and that's the big advantage, actually, they are so specific, much better than many of the organic compounds that are around. Many of the available organic inhibitors, staurosporine-derived derivatives, they aren't quite that specific. They are more limited, and they might inhibit a whole group of kinases. And of course, if you want to study one particular cellular process, it's nice if you can inhibit exactly the one kinase in your process and not too many others. So in that sense, this group of compounds is really, really useful. It may be useful in the future for, as a drug, but it's already used in biotechnology work or cellular biology work for fundamental studies. This is intended to give you just a brief indication of the chemistry that you can do to make these compounds, and you don't need to understand all the steps here. I'm just going to introduce to you a couple of compounds that you can make, and you can see this slide here has this table, but there are quite a few more A, B, C. So on this slide, you certainly have 20 different compounds, at least, that all can be made more or less by the same route. And then you can, when you have those 20, you can go for your screening in your kinase assay, and I can guarantee you will find some really good and quite specific hits. There are two main points here I want to point out in the synthesis. You synthesize the ligand, and of course we already saw that, yes, we need to modify the ligand here, put some groups on either on this side here, or maybe even up here. There are many different ways. So you make the ligand first. This is organic synthesis. You need a trained organic synthetic chemist in order to make many derivatives of this. You add with iridium chloride cyclooctadiene dimer. So this is a simple addition reaction. And then you get an iridium compound in oxidation state +1, which is square planar. So this is the key starting material. Organic chemistry, iridium precursor, iridium-1. And then you do an oxidative addition reaction, and you go from iridium-1 square planar to iridium-free octahedral. For example, here you use iodine, so you add 2 iodine atoms, going from iridium-2 to iridium-free formally. Here you add methyl iodide, so you have a methyl and an iodine group, and then you can do substitution reaction, for example, of the iodide. You can substitute with let's say methyl Grignard or any other reagent, you get another group Y here. You can use other oxidation reagents. Here you see chloro-trifluoromethyl, for example. You can think of many, many other oxidation reagents, and then you would get the appropriate iridium-free compound. So step 1 is coordination iridium-1 to the ligand. Step 2 is oxidation by a suitable oxidation reagent going to the octahedral iridium-free compound. And it's basically as easy as that, almost. So this is where iridium chemistry comes in really handy. 2 steps to go to octahedral iridium compounds of quite significant flexibility. When you look at this, 20 different compounds, certainly on the slide, 2 key reactions, and the rest is just cleverly playing around with substituents and using, the right type of conditions. Okay? All right, okay. These compounds, here is an example what they actually do. This is an animal experiment on a very primitive animal. This is a fluorescence picture of zebrafish. Zebrafish is sort of small animals that, as the name implies, fish. The good thing is that the zebrafish embryo, the small ones, they're actually transparent. So you can look through the zebrafish when they're young. And the good thing is you can see, this is what is used here, you can see their blood vessels. And one critical thing is that you can see how the formation of new blood vessels, how this works. And you can imagine, we had these pictures, the tumor that needs the blood vessel in order to get its oxygen and its nutrients. And therefore, when you have a tumor somewhere and it starts growing, it will actually form new blood vessels around it, the tumor, in order to get enough supply of nutrients and oxygen. Yeah? So the tumor will induce growth of blood vessels around it. And if you can inhibit this, then you can starve the tumor. Yeah? Simply by cutting off water supply and electricity, basically. You starve, the tumor that doesn't get this anymore. And you cannot, you can, but it's not a very pleasant way. You can't really investigate this in humans or in mice or so. But you can easily and nicely investigate this in zebrafish because, as I said, zebrafish is transparent. You can color the blood vessels by a fluorescent dye, like this green dye you're seeing here. And you can really easily see all the blood vessels of the zebrafish. Now you feed this zebrafish this compound here, that is a kinase inhibitor, that is obviously involved in the maintenance of lymphatic vessel and the development of a new cardiovascular system. And if you feed the zebrafish this compound, then you see this, what is probably looks a bit like the spine, doesn't it? Here, really nice, lots of blood vessels. You can see here, there is a break here, a break here, a break here. So this nice sort of spine-like supply system suddenly is shut off, and the blood vessels here haven't really formed, really. Yeah? So this gives you an idea what you can do. And this zebrafish experiment has been used in many, many other studies as well as a model of the formation of new blood vessels. Yeah? So this is called angiogenesis, is when you form new blood vessels. So anti-angiogenesis means blockage of formation of new blood vessels. It's a viable strategy in anti-tumor research, obviously. Yeah, it's like starving the tumor. Rather than killing it directly, you're starving it. So indirectly, it will die after some time. All right. Here you can see the same. You can see these sort of formed, this control. It's sort of the color here forms new blood vessels. And this is the kind of investigations that people do. You can see here is a change. There used to be something here, and then under treatment, it's really gone. So this is the kind of experiments that people do here. And this kinase, this one you have seen, is involved in the formation of new blood vessels. But as I said, there are 499 different ones or additional ones, and they all have all kinds of different functions. So as often, it's a question of finding the right assay and identifying the right question. And then you can, find the right assay and you can investigate whatever your question was. Okay, we'll go through some more enzymes where organometallic or metal complexes have been used. Maybe not quite in such depth, but I think the story of kinase inhibitors, I actually really like this very much because it's a really original research program. You can clearly see where metal complexes have a benefit. That is to make these very complicated structures very straightforwardly. I mean, think of the synthesis of the iridium compound, yeah? One slide, 25 different compounds, or 20 or so, whatever the number was. That's really quite straightforward. So I really like this program, and it has furnished spectacular results. Okay, let's move on to a different class of enzymes, the so-called telomerases. Telomers are very important, and those have to do actually with the DNA again. But it's at the end of the DNA because there is so-called a telomere region, which consists of repetitive DNA fragments, and basically it protects the DNA from degradation. So you have this long DNA strand, and DNA is always degraded and is built up again, and sort of it's eaten up from the end. And if that sort of end eats up more and more and more, it eats into your actual DNA, and then things start going wrong. So this is basically a process of aging. This is to determine how old is a certain cell or how old is a certain organism. When you are very young, you have very long telomeres. And then in your lifetime, they are sort of eaten up from the end. And the shorter they get, the older you are in a way. And at some stage, you will really be affected, and some things may not be operational anymore. That partly has to do with the fact that, the telomeres are eaten up, and whatever, the degradation starts getting into your real DNA. Of course, you can say in order to fight this, yeah, you can have an enzyme called telomerase, and this actually adds hexameric units to the end of your DNA. So telomerase is like an anti-aging drug. Yeah? It adds additional DNA always to the end of your DNA, to the telomerase, in order to suggest that the cell is actually younger than it is. So it's like some of these creams which you buy and put on your eyes to remove the wrinkles, to make you look younger than you are. So telomerase is the enzyme that makes cells look younger than they are. And of course you can imagine, if you have a tumor cell, very often telomerases are overexpressed, so they work like hell. Yeah? They add more and more and more and more DNA to the telomerase, so the telomerase, the cell that has these, they will never die, because the DNA always looks super young. Yeah? So it's like immortality, believing that you are 20 all your life and even longer. So telomerase does exactly that. Yeah, it pretends that the cell who has a telomerase overexpressed pretends to be at the age of 20 all the life. And of course that's bad for a tumor cell, because the tumor cell, you want to die. Your hope is that sooner or later tumor cells get older and they die by themselves. And this is a mechanism of preventing this. So, this is it's good news for the tumor cell, because, the tumor cell feels 20 all the time, but it's bad news for your organism because, they never die. So if we can inhibit the telomerase, then we might be able to drive the tumor into apoptosis, into cell death, at least after some time. When they split and split and split, then they don't split any further because they lose their telomerase, and then that's the end of it. And there have been many telomerase inhibitors have been tried. I find this particular group of compounds quite interesting. Those are osmium compounds. Osmium, not toxic, low-valent osmium in this case. And these are sort of osmium carbonyl type complexes, very well known in organometallic chemistry. They have some specific ligands, for example, the sulfonated phosphonium ligand, so they're actually water-soluble, or you can have ammonium groups here that make it water-soluble, but otherwise it's an triosmium carbonyl kind of core, maybe with some auxiliary ligands here. But overall, these are very classic organometallic compounds, if you like, and they were shown to be quite potent telomerase inhibitors. Okay, there are some problems associated with these. One is that they are ineffective in vitro, a different DNA polymerase, and also, they weren't really effective on this MCF7 cancer cell lines. And moreover, I told you not every osmium compound is toxic. Unfortunately, these here were actually acute cytotoxic. And partly, this was probably because if you look at the structure, they're quite lipophilic, and they actually accumulate in the cell membrane. And the telomerase, of course, because it's a DNA enzyme, is of course in the nucleus of the cell, because it has to work on the nuclear DNA all the time. So if your compound gets stuck in the membrane, that's not good news, because you want the compound to enter the nucleus and not be stuck in the membrane. So we really need to work on this. And something that people have tried is cationic porphyrins here. For example, this kind of molecule where you have these pyridinium groups here at the outset, and you can already guess, this compound by itself binds already to telomerase and inhibits growth, but it's not a very good compound by itself. But you can already guess, if you just look at the structure, this looks like a porphyrin, and this looks like a metal binding site in the middle, doesn't it? It's like iron in hemoglobin, for example. Iron in heme, you really want to put a metal in here. It's just asking for it. Okay, there's some more information here. We don't really have to read all this out. But octahedral metal complexes, you were made. They were actually less active here, but I think there is an example here. Here, the zinc complex, exactly. The zinc complex here turns out to be actually quite potent, and you can see it binds to these G quadruplex DNA, which are part of the telomeres, and you can see it has nanomolar affinity. So, it should be the other way around. It's smaller than 2 nanomolar. So that is very, very high affinity suddenly. And you can see here, it's sort of, this really flat structure. When you think of quadruplex DNA, where the DNA is stacked like this, and this molecule here really nicely inserts into the stacked DNA. It's very flat. It has some positively charged groups here that interact with the phosphate groups. It has a zinc in the middle. That is also good. The charge in the middle is helpful. So this is kind of a compound that has very, very high affinity to telomerase. In this case, it's a coordination type compound, unlike the osmium organometallic compounds that we had previously. But there's quite a lot of research in this area, and if I look at, sort of recent issues of Dalton, I would say at least once in a month there is a paper in Dalton on compounds that work as telomerase or G quadruplex inhibitors of one way or another. Another class of important enzymes are so-called zinc finger proteins. Again, they work on DNA. They are involved in DNA transcription and the various repair functions. So again, you can see if we can inhibit one of those, then certainly we can, for example, we can stop DNA repair, which would be a very good idea. Now, if DNA is damaged, say cisplatin binding, DNA repair comes in, cuts out the platinum, cell continues to live. If we can stop this, then we might have a good way of actually killing the cell permanently without possibility for repair. And they're also involved in other things like HIV. I will talk about HIV and AIDS at the very end, I think, of the next lecture, so some more here. And again, there are compounds that were implicated as being zinc finger protein inhibitors, and strange enough, you see here, those are compounds we know really well. They have a cisplatin-like structure almost, but they're not quite cisplatin. They have only 3 nitrogen ligands and 1 chloride. So those are like monofunctional platinum compounds. Clearly, a compound like this cannot do the classical DNA crosslink because there is only one open coordination site here. So it can only bind to one target at a time. And therefore, these compounds aren't really active on DNA so much, but they have used this open site and they bind to proteins. And if they're just made like these ones here, they're really made to bind into these zinc fingers. And they also have to do with DNA transcription, but in a different way. Not by binding to DNA, but by binding to an enzyme that helps in DNA transcription. So yes, there is DNA-related activity, but in reality, it's not on the DNA, but on an enzyme. We have another compound here. This is kind of a classic compound that was made already in the 1960s. These compounds were actually invented, type of coordination compounds were invented by Schautzer and others as mimics of vitamin B12. Remember Monday afternoon lecture. This compound of this class has activity similar to vitamin B12. You can make the methyl compound or the adenosyl compound. You can get radical chemistry out of these compounds very nicely. And they have Okay, everybody is ready for the second round. I promised you we would switch targets, not so much compound, but switch targets. We are now talking about infectious diseases and how we can use metal complexes to potentially cure them. There was one, again, as all the days, there was one very interesting thought from one of you that I would like to take up. And the question was, especially if you look at, the many, many compounds you have seen here, and I said before, there were hundreds or maybe thousands of platinum compounds were made and screened. One of you said, well, isn't that a waste of time and money and probably mice and maybe higher animals if we have to make all those compounds and screen them in order to find the best one? Isn't there a better way of doing this and, some clever way of finding predicting activity before you actually have to make the compound and before you have to sacrifice the animals? The answer to this is yes, there is. It was on one of the very first slides, and it has to do with computer science. People try to do structure-activity relationship, as we have discussed, for example, in the Megas compounds. This is a very good case of structure-activity relationship, where you say, we'll put a chloride atom here in order to fill this pocket. Yeah, this is structure-activity relationship. Higher activity by a change in structure. Now you can do this, you can train your computer to do this. You can fit the crystal structure that was derived from the kinase into your computer, let the computer predict what the size of the pocket is, and then suggest a molecule that would fit into it. And if you can put numbers to it, for example, predict binding affinity, this is called quantitative structure-activity relationship. So QSAR, QSAR. Yeah? SAR is just structure-activity relationship. Quantitative, meaning putting numbers to it, predicting this is better than this one, and this is twice as good as this one. This is called QSAR. There was a big hype on QSAR maybe 20 years ago in the 1990s. When many crystal structures of targets became available, when computers were beginning to be powerful enough to really predict molecules and crunch large number of data, when this came up, I can tell you, all the big drug companies were investing heavily in computer equipment and more so in computer scientists. And I would, really predict a lot of Indians were hired as computer scientists in that time. The fact that even today, in 2017, 20 or 30 years after or so, 20 years after, we are still making molecules and screening molecules, what does that tell you about the success of the QSAR program? It wasn't very good. I mean, that is not to say, don't try. You get some important information from it, certainly. It certainly helped limit certain spaces, or limit programs or drive programs in a certain direction. But it didn't fulfill the hope that we don't need to make hundreds of compounds anymore, but only make maybe one or two compounds and then have the perfect hit already. Yeah? That's unfortunately not the case. We still have to make many, many compounds and screen because many things cannot be predicted in a computer. You can't predict the best possible compound to fill a certain enzyme pocket. That you can do. That is no problem. I mean, if you have enough money and you have a clever computer scientist, you can tell them to do it for you. But to predict the action from, taking a compound as a pill to it actually curing a disease is much, 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 much more complex. And it's way too complex currently for a computer to model. Maybe if we have even better computer, and maybe if we have quantum computing, and maybe if we have, smarter algorithms invented by smart Indians in Bangalore, I don't know. It may well be. I'm quite hopeful actually that there is important information that you can get, and it means one should work on it. Yeah, I'm not against it. I'm just saying the whole process, from the pill through your body to the target to actually healing the disease seems to be way, way, way too complicated currently to actually model it. But that is not to say you shouldn't try. I mean, maybe one of you has a really smart idea to advance this field, to take certain effects into account that have never been taken into account before and to advance this knowledge. So, I'm highly encouraged. Look at this and think about it. If you like computer science, you like medicinal chemistry, there is lots of things that certainly can still be done. And it's worthwhile because you are saving a lot of time of your colleagues in the lab and you're potentially saving a lot of animals. So, go ahead. Okay. So again, a very, very good remark. I'm really, really grateful for these discussions in the breaks and after the lecture. So, it also teaches me a lot of things. You come up with very good questions, really. And, it's like, makes you think, like, yeah, why not, actually? Yeah, good point. All right, let's switch to infectious diseases. We've been talking about cancer a lot. Of course, cancer is very, very bad. And if you have somebody in your family who has died from cancer, as I said, my grandmother died from cancer. That is certainly very bad and certainly very sad. The reality is that infectious diseases as a whole are a much greater, not a much greater risk, but they are much more grave threat to mankind, meaning many more people are dying worldwide from infectious diseases than from cancer. So total, if you take together bacterial infections, malaria, tuberculosis is coming back, tropical diseases like leishmaniasis and so on, dengue, yellow fever, things like this. If you take all those together, you can already see, there'll be 10 times more people dying from those than from, for example, from cancer. We will also briefly talk about HIV and AIDS, viral infections. This, in fact, will not be a large part of this lecture because there's a lot of research. And, of course, AIDS and HIV have received a lot of attention. There aren't really many metal complexes that are instrumental here. But some others, we have quite some nice successes. So I won't talk about all of those here. Don't be afraid. I'll pick examples and tell you stories. And then there's also, again, more information on some of the slides. I may not mention every single fact that is on the slides, but never mind. I promised you this timeline would come up again and again and again. Well, here it is again, and here is what you need to look at. We can look back easily 200, 250 years, and you see that the first, what I would call clinical studies, were initiated already 200 years ago. In this case, it was on bismuth compound, yet another metal. We'll touch this. And then Robert Koch, as I said, already mentioned these gold compounds here, the gold cyano compounds as antibacterial were really well-defined compounds here. And then the first structure activity was actually done SAR. We just discussed this here. The first time that it was really, really systematically done was actually on an arsenic compound, and this is about 100 years ago. And we'll briefly talk about all these cases here. So you just see the work on infectious diseases goes actually back way before, people thought about cancer and fancy things like kinase targeting and so on. So this is really very basic. This was the quote which I've already seen before. I just put it in here because it fits so nicely. Robert Koch, a German scientist actually working on bacterial infections in the 1890s, said that cyano-gold compounds here were the most active ones, and you see here this was published in 1890 from this Congress. It was Congress proceedings, and it had this hint towards gold compounds already in it. The first compound that was actually really explored and tested through this structure-activity relationship is, as I said, is this arsenic compound that is related to the name of Paul Ehrlich, and I'll show you a picture in a second. The background here is that syphilis is a sexually transmitted disease, and at the time, this is 200 years ago, of course, people didn't really know about how diseases were transmitted through, bacteria or whatever. This knowledge just came up with people like Robert Koch, whose name you've just seen. And, of course, people didn't use condoms at the time and so on, and all the things that, might help you prevent such diseases nowadays. So this particular disease, syphilis, is actually, it's from a gram-negative bacterium called Treponema pallidum. It enters the blood through the epithelium, and if it's not treated, it will finally, this bacterium will end up in your brain, in your nerves, and it will cause blindness and death. So it was a deadly disease if untreated, and nobody had a cure at the time. It was called, interestingly, the French soldiers' disease. I don't know whether that is to suggest that the French soldiers were particularly active, not just as soldiers, but in other fields as well. I don't know. Maybe it should. I don't know. So this compound here, called arsphenamine salvarsan, is the first compound that was really effective in curing syphilis here, so to treat syphilis when the person, the soldier, the normal person, were actually affected. It is the first compound ever where somebody had worked out a structure-activity relationship, and you note, of course, that this was an inorganic compound, and it's a main group inorganic compound, if you like. The work was done in the lab of Paul Ehrlich. This is a picture of Ehrlich, sort of like from, I think it's actually from the Nobel website, because he won the Nobel Prize for Medicine in 1908. This was just after the discovery of this compound salvarsan. At the time, there was no thing with patent and patent issues and so on. That wasn't there. So as soon as he had discovered this compound, he already spoke about it and spoke about his, the animal experiments they had done and the experiments on humans they had done. So very quickly after the discovery, he received the Nobel Prize together with a Russian researcher. At the time, the Nobel Committee wrote in recognition of their work on immunity. So this was still considered, there wasn't something like microbiology or, fields that we have today. That wasn't really known. All this was known as immunity, so as if the immune system would cure the disease or so. Because of this discovery, it was actually the first, systematically explored field where a viable drug resulted. Paul Ehrlich is very often considered the father of chemotherapy. And the word, you may have heard the saying of the magic bullet. Who of you has heard of the magic bullet in terms of drugs? It's in some textbooks sometimes. What it means is that the drug should hit only the bacterium in this case and not the human. So this was the magic in it. It shouldn't kill everything. It should just kill the disease and not the human. And of course, this is in the early days when they still knew that there was a parasite, but all the things like enzyme inhibition and specificity of enzyme inhibition, all these concepts weren't yet known. Was not yet known. But the magic bullet concept referred to killing the parasite and not the human. And today we know we can transfer this and say finding something that is very specific for a certain enzyme in terms of enzyme inhibition, for example, or a certain molecular target. But this phrase of the magic bullet was coined by this guy, Paul Ehrlich. In fact, he was so famous, he was on this is a German 200 Deutschmark note. So you can see it should be somewhere on the side. So it's out of, it's not currency anymore because we have the euro. But this was until 2000. This was legal tender. And that is Ehrlich, as you can see, on this 200 Deutschmark note. What you will also see, if you look very closely, here is a chemical structure. Yeah, there are phenyl rings here. It's a 6-membered ring. Can you see it? Just about. So there is a chemical structure on a note on money. I mean, I think that's totally amazing. Yeah, I don't think it would make it today anymore. But this is when people still believed in science and not in fake news. So it's really when it was on there. So I think that's quite remarkable. You'll see actually that there is actually a lot of truth in this 6-membered ring here. I'll tell you why. Let's briefly look at this compound here. We'll come to the 6-membered ring in a second, okay? Let's quickly talk about the compound here, and then we look at the chemistry behind it. This compound was called the 606 compound that was made in his lab. So you can see it was a very extensive structure-activity relationship, making 600 compounds until you have a hit. Number 606 was the one that was most active. This is the structure as it is formulated. We'll come back to this arsenic-arsenic double bond in a second. It turns out that this hydrochloride, which is prepared, is too toxic, so the salvarsan is actually the free base. The structure of this compound for a long time had not been known. It was made, it was prepared. We actually made it in my lab. The original prep works quite well. It's quite remarkable. You can go back, into the literature 100 years, you find how to make this. It's a bit of a, a sort of magic, but it works nicely. You isolate a compound just as described, melting point, everything is fine. We go to the structure in just a second, but later on it was discovered that, again, this is a prodrug, and the actual active agent is the oxide of this. So this is oxidized in air, in the blood, in your body, and you get the arsenic oxide. That is the actual active ingredients. You can modify this amino group slightly, and it gives you a better active compound. The interesting thing from a chemistry point of view, I think if you look at those today, is that even today, we do not know what the exact mode of action of salvarsan is. It's a relatively simple compound, but nobody has identified the molecular target or the targets where this compound bound. Isn't that surprising? It's 100 years old. It's such an important compound. Nobody knows. Also, the structure of this compound, it was very often drawn with a double bond. Also by Ehrlich at the time, he draws this compound with a double bond. The structure of this compound for a very long time was not really known. It was known it's a mixture of some things, but nobody really knew what it is. And people suggested some cyclic compound. And the compound which you see here on the ring is actually a 6-membered ring, but this is just a phenyl ring. So if you imagine you remove the amino groups and the OH, and it's just a phenyl ring, and you look at this compound, so phenylarsane if you like, this actually forms a hexameric ring, and you get this structure, and this is actually a crystal structure of the phenyl derivative of salvarsan. So it's not quite salvarsan, it's more simple compound, but this is confirmed by X-ray crystal structure. So people thought, oh, maybe this one here, N should be 6. And of course, you all know that this is nonsense, because your textbook chemistry, first or second year main group chemistry, tells you that a double bond between two arsenic atoms is very, very unlikely. Yeah? If then you have to have large substituents here to shield the double bond from dimerization or oligomerization. So you know this formulation is chemical nonsense, and what it is was actually discovered only about 10 years ago. So 100 years after this compound was made, people actually looked at the structure, in this case by mass spectrometry, and it was elucidated that it is a mixture of cyclic compounds, and the main components by mass spectrometry are this 3-membered ring and the 5-membered ring. So unlike the phenylarsane, where it's just a 6-membered ring, there is a mixture here, and the main compounds being those two here. And this is a paper that in Angewandte Chemie that has been cited quite often. I guess if you had a plan to investigate the mode of action of this compound, you would be equally highly cited. Okay, so that is salvarsan. As I said, we don't know much about its activity, so let's go to some other very, very famous, very old compounds, and this is the group of bismuth compounds. We already saw that bismuth has been explored systematically 200 years ago. Bismuth was known to be a good treatment for stomach diseases, which we call more scientifically peptic ulcers. So it has to do with problems in your stomach, and these ulcers are very often caused by this gram-negative bacterium, Helicobacter pylori, and what Helicobacter pylori does is actually very, very, very clever. In your stomach, you have the epithelial cells, which is the cell wall, and your stomach, as you know, is very acidic. Yeah, it's like pH 2 or something, 1.5, 2, 2.5. So in order to protect your cells, there is this layer of a very gel-like thing on it that's called the muscus, which is sort of protecting your cells. So it's on the inner side, and it protects the cells from the very acidic environment. So it's like a protective coating, if you want. And what this bacterium, Helicobacter pylori, does is when it becomes pathogenic, in fact, most of us will probably have it in our stomachs, and it's not doing anything. It's not harmful at all. But if it becomes harmful for whatever reason, it uses an enzyme, so-called urease, which produces ammonia, and then it excretes the ammonia here. So by this adding this ammonia, it lowers the pH locally, very locally. At this point here, there is suddenly a high concentration of ammonia, which means that it becomes basic. This gel here, this mucus, is of course then not gel-like anymore, but it becomes much softer, and then the bacterium can enter through its own pH sink, if you like, can enter into the mucus, and it can reach this epithelial cell, the skin, and then it will form a colony. And then it starts growing on this colony, and it protects itself from the acid in the stomach, by producing a lot of ammonia. So to have a nice neutral environment. Yeah? This is actually pretty clever. Yeah? It's a pretty nifty way of doing it. It's a nickel-containing enzyme that simply hydrolyzes urea in order to produce ammonia. So in most cases, as I said, it's not harmful, but then if the bacterium reaches the skin here, the epithelial cells, it forms colonies, and the colonies start growing, then you get ulcers and you get stomach diseases, and this is pretty bad for you. And it has been known, again, more than 200 years ago, that preparations that contain bismuth can be easily used, can be very well used to treat these stomach disorders. And that's been known for a long time, even before people knew what exactly was in their preparation. They just knew if I take certain salts and I add this and I prepare it in this particular way, it helps to treat, to clear these stomach disorders. And today we know from these preparations that they contain bismuth and things like, for example, salicylic acid, which is also a plant ingredient. These preparations are remarkably non-toxic, partly because the bismuth, if you take it, if you eat it, because it reaches your stomach, it's just excreted. Whatever isn't needed is just excreted. Bismuth in this forms rarely ever gets into your blood, so there's actually no problem. It's sort of like a topical thing. It goes through your stomach, through your intestines, and leaves your body right away what isn't used. And of course, if you think now back in your chemistry, bismuth 3+, which is the active, sort of active compound in it, it's a main group element. Ligand exchange will be rapid. There is no ligand field stabilization energy because it's only S and P electrons, no D electrons, no ligand field. So ligand exchange will be rapid. There is no preferred geometry. Everything will be pH dependent. You will probably have carboxylate ligand, for example, or carboxylate or alcoholate or water or hydroxy ligand. So everything will be pH dependent, rapid ligand exchange, and you can imagine this is going to be horribly difficult. In fact, the active compounds probably contain bismuth 3+ in the form of some large clusters, and I'll show you some examples that people have actually x-rayed. The most widely used drug even today is what is called bismuth subsalicylate. So this is salicylic acid mixed with bismuth salts, and as you can see, it's not a 1-to-1 ratio or what is a 2-to-3 ratio, but it's some sort of like not quite enough salicylic acid. So it's some weird preparation. It's, for example, it's called Pepto-Bismol and things like this. You can actually buy it over the counter. I was actually planning, I forgot to do that, I'm sorry. I was planning to go to some random, these on the street. You sometimes have these pharmacy type shops in India. I was planning to go to one and ask for bismuth drugs, and I'm pretty sure I would get something. Either this one, or there are a couple of other ones. My main problem would be to communicate with the person and tell, I can only say I want a bismuth drug. And that person will probably look at me and say, what do you want? So, but I'm going to try. I mean, I thought of it when I came here. I thought I should do this experiment. And I thought it would be nice to show it to you. I can try tomorrow, maybe. Maybe we'll manage. See how far I got with my Hindi. Okay. All right, okay. So these are over-the-counter drugs that don't even need a prescription because they're basically not very toxic and they help you, rapid relief of heartburn and things like this. These are some of the structures that, this is now scientific, it's sort of like real preparation of compounds. People have made bismuth oxocluster with salicylic acid with additional auxiliary ligands. You can see here, this is like salicylic acid kind of derivative. what is this? It's a bismuth 9, oxygen 7, 13 salicylic acid, and some deprotonated acetate again. So these become very, very big, very, very complicated clusters. Chances are that there are equilibrium arrows here everywhere, and the story continues, and there are many, many more on this other side. So many, many different species, I think, will form, and they'll be in equilibrium. So the question is, how do these things now actually work if they're all in equilibrium? And I guess people think, I don't think it's on here, is it? So what people think is that these simply form these clusters, and I think, wait, it must be somewhere here. Yeah, the mechanism of action, here we are. I think those two slides should actually be changed. So the mechanism of action of these compounds is still not very clear, but what people think is that these bismuth cluster actually might form a precipitate, so on the ulcer crater, so they basically facilitate the healing, so it's like a micro coating, that is on the top of these ulcers. People also think, and this is now very chemical, they think that bismuth might replace nickel in the active site. So it's a 3 plus bismuth. It might have quite a good affinity actually for the bismuth for the nickel binding site. And if that happens, then of course the chemistry doesn't quite work anymore, because bismuth, of course, is totally different from nickel. So that will stop the urease from working. It has also been suggested that it is not the replacement of the metal in the active site, but some of these clusters or some of these compounds might actually block the entrance where the urea comes in and then binds to the nickel. And if there's a big block of, a bismuth cluster, then the urea can't get in anymore, and then the bacterium cannot produce ammonia anymore, and slowly it gets more acidic, and then the bacterium dies in the acidic environment. So that's some of the thoughts that people had. So these are actually quite sort of chemical suggestions. It's kind of difficult to prove, as I said, because you don't even know what the active compound is. It's certainly a mixture of some things. You see here there are many more compounds that are in investigation that you can buy. So this is colloidal bismuth citrate called dinol. Here this is another compound that has been used, and then there are a few more bismuth compounds. These have sulfonated ligands, so there's more. You see here, this is like salicylic acid, but with a sulfonate group here, so it's more water soluble. This one here has phenyl groups on it, so it's like an organometallic phenyl bismuth with some sort of other ligands here on it. So people are experimenting with these compounds. There are nowadays, I would say, a small handful of groups in the world, maybe 5 or 6 or so, that work constantly on these bismuth compounds and try to find new ones, better ones. As far as I know, my friend Phil Andrews, for example, from Australia, is one of those in Melbourne. As far as I know, I don't think they have got anything that is, better on the market. In fact, the things that are on the market are actually already pretty good. I think we discussed this briefly during coffee before the lecture. Today, there is also an alternative treatment possible, of course, because the bacterium, Helicobacter pylori is simply a bacterium, so you can use normal antibiotics to actually kill the bacterium, and that's what people very often do. When you have these stomach disorders and it's really down to Helicobacter pylori infection, you're basically given normal antibiotics. The main problem is that the antibiotic must reach the Helicobacter, which is on your stomach, and of course you don't want to kill the antibiotic, but you really need this to get to the Helicobacter site, and that's the main problem. Okay. So that's the ulcer and the bismuth story, which is quite interesting. In terms of structure-activity relationship, there isn't so much to learn here, but in terms of, clever chemistry and some clever activity, that's actually a pretty good example. I would like to move on to some tropical diseases. For example, this one here, leishmaniasis, is again one of the diseases. It might actually be, I guess it's a topic also in India. Huh? You have many patients, people suffering from leishmaniasis? Yeah. I read here it's India, Bangladesh, so it's like this sort of tropical regions. The disease is transmitted by the bite of a female sandfly, so very simple thing, and it's caused by this protozoan So it's like a monocellular organism. This parasite is called Trypanosomatidae family. So there are different ones, but they're all pretty bad. If you are infected by this one here, by the really bad one, then it affects your liver, your spleen, and if you are not treated, the mortality rate is actually very high, 95%. So you really, really, really have to do something about this. You have to treat this. Otherwise, there is a good chance that you are going to die. So there is definitely something you have to do about it. Basically, this is this protozoan. It's like a worm-like thing. So you really need drugs that are against these anti-leishmanial drugs. And the astonishing thing, if you look up the literature, then you find that even today, it's these antimony-containing drugs that are still the most common on the market. I mean, there are modern antibiotic-type treatments, but there aren't really many under development. And I guess this comes back to my first lecture again. If you see where those cases appear, I mean, don't take India and Brazil, but if you see Sudan, Ethiopia, Bangladesh is also doing better. But these are some of the poorest countries in the world. So the problem is that there isn't actually so much money here to develop new drugs. As I said, don't count Brazil and India. I think the situation is definitely improving. There are also more research now on these drugs, on anti-leishmanial drugs. So the situation is definitely improving. But it's quite amazing to see that these compounds here, these drugs, were discovered in the 1940s, basically following, the Ehrlich thing. Ehrlich was working on arsenic, and he was good against syphilis. So clearly people, went in the periodic table up and down and left and right, and you arrive at bismuth, and you arrive at antimony relatively quickly if you start from arsenic. Yeah, it's right next to arsenic in the periodic table, or below arsenic, actually. So this is kind of an obvious way to look, and they found, preparations like this sort of simple organic ligands called meglumine, antimonate is active, or this gluconate derivative here of antimony, both are active against leishmaniasis. And they were discovered already 70 years ago, and they're still some of the most commonly used compounds. So think of this again. Many of what we said for bismuth is true here as well. It's antimony 5. It wants to be octahedral, but it's a main group element, so there's no crystal field stabilization energy. You can have rapid ligand exchange. There is no really preferred geometry, so you can already guess there will probably be some follow-up chemistry, hydrolysis of ligands, ligand exchange reactions, and so on and so on. It's really difficult to study what the active species here in solution could be. That's really not clear at all. It's probably, again, a mixture of some higher aggregates and so on and so on. What we do know, though, is that this is just a prodrug, and really, again, these antimony 5 compounds are reduced to antimony 3, and this is usually considered by most people to be the active species. And this, again, is a question, I think some of you asked this yesterday. We were talking about thioredoxin reductase, and we were also talking about the question of targeting. How can we make things specific for certain organisms or for certain species or so? Well, here's a way how you can do that. And thioredoxin reductase is a system that we have in humans. Now, this trypanosome kind of family of organisms, they have something that is called trypanothione reductase, and it's called abbreviated T and TR, so it's much easier to pronounce. Okay? So this trypanothione thing here is the equivalent of thioredoxin, but it's now particular for these organisms, and that's maybe why the antimony works on these and on this system here, but it doesn't work on the human thioredoxin reductase system. And that's what people believe here. There's an opportunity for species-specific targeting because they have their own particular redox system. And here's a crystal structure of the antimony-free preparation with reduced trypanothione reductase. And you can see the antimony binds very nicely here. It binds to 2-cysteine residue and to this histidine, and there may be some additional ligands here, a threonine in the background, and maybe 2 water molecules that aren't shown. So this here basically binds to those two cysteine. It blocks the cysteines because the cysteines want to form a disulfide bond during the catalytic cycle. And if the antimony is binding here, they can't form this disulfide bond anymore because the antimony is in the way. And then, the activity of this particular enzyme is terminally blocked. So it's inhibited forever until somebody removes the antimony. And that is what people believe is the mode of action. And as you can see, there is an opportunity here for specific targeting of this particular system in the bugs, in the parasites. If you look at this is kind of familiar to you. It reminds you, for example, of the ruthenium compounds we looked at. It looks as if all the ligands on the antimony are lost in the process. On the other hand, if you look back to the structures, that's not surprising because 2 of 3 of them were hydroxide or water anyway, and then there is only some alcoholate ligands. So none of these are very strong, very stable ligands. So people have tried to work on this, and for example, some experimental drugs are all phenyl antimony kind of compounds. So like 2 phenyl rings here or so. So people have tried to work on these kind of compounds. But you see that also some of these ruthenium compounds reminds you of the ruthenium arene compounds or vanadyl compounds of this type have also been studied. And if you look at the variety of these molecules, if they're all active, well, that already tells you structure-activity relationship, very difficult, If all these are active, difficult to say what kind of parameters you need to get an active compound. It's very much a trial and error field still. Yeah? What you want to do here, which way you want to progress, very much trial and error. Okay. I have one more interesting story that I would like to tell you. It's about malaria. And I think arguably you all will agree that malaria really is a very serious disease. I found those numbers from the World Health Organization. It is about 650, it says here, has gone up since. There is certainly in the order of 750,000 people or so in a year are actually dying from malarial infections. This is a huge number. Yeah. And certainly hundreds of millions of people worldwide are affected, and out of those, as you can see, a certain percentage die every year. It's largely children that suffer because they don't quite have the immune system. They're exposed. They're more difficult to treat for various reasons. So it is really a severe problem again, and you all know this. Maybe not so much in India anymore because treatment is available. India is not a very poor country anymore, but it's clearly in sort of like southern, less developed region where this is a main problem. You know about the parasite that causes malaria. It's this Plasmodium species here, and the Plasmodium enters the body when you got a bite from the Anopheles mosquito. And then the whole cycle of this parasite sets in. Some of the problems here is, and if left untreated, of course, then the merozytes reach the brain and you die from coma and death. Part of why malaria is so difficult to treat is in this complicated life cycle of the parasite. And this is taken from the Center for Disease Control and Prevention in the US. This is the human, this is the mosquito life cycle. And you can see that there are several stages which the mosquito undergoes, and it goes in the liver and it goes in the human blood. And all these cycles are connected, and in all those cycles the parasite actually is in a different form. So it's really difficult to target, for example, when the parasite is in the liver, it is a totally different target than when the parasite is in the blood. In fact, when the parasite is in the blood in large numbers, it's actually already too late almost. You would like to eradicate either the mosquito or the parasite in the mosquito, but unfortunately here again the parasite is in a different stage. It's called an oocyte, so it's in here and it's difficult. But the thing is, the parasite actually enters when you got a bite. It enters your liver first. There's some liver stages here. The parasite replicates. You get many more. You got in this schizontic state, and then the whole thing bursts apart, and these schizons are actually released in your blood, and then they form something else, which is called, where is the merocyte? Should be here. I can't quite see it where it is. And that sort of replicates again, and replicates and replicates, so it multiplies in your blood again, but it's a different cycle from the cycle here in the liver. So this is really quite tricky, quite difficult to cure. The main site of attack is actually the metabolism of the parasite when it's in the liver. And what Plasmodium does, it degrades hemoglobin to obtain amino acids. So that's a sort of parasitic way, truly parasite. It takes from the red blood cells of the host, it takes the hemoglobin, chews up the hemoglobin, recycles the amino acid for its own metabolism. So it cannot produce amino acid itself. It needs them from the host, from hemoglobin. And then what it doesn't need is the heme, the core, the tetraporphyrin with the iron in the middle. So that is kind of discarded, and the amino acids from the globin are used. And what happens to the heme? It accumulates, and this is the parasite. It's so-called the food vacuole of the parasite. So this is basically the stomach of the parasite, if you want, where heme is degraded, amino acids are used for the metabolism of the parasite, and then the heme, iron and porphyrin, sort of precipitates, and it forms what is called hemozoin. And the hemozoin, if there's like a lot of heme stacked onto each other, it's actually called the malaria pigment because it's dark blue colored, and you can actually see this when you have infected blood cells. You can actually see the parasite inside and those dark blue precipitates, which is the hemozoin. Yeah? But this is quite okay for the parasite. It's like sort of the waste side for the heme, and then the parasite uses the amino acid, it multiplies, and then in the end, of course, the parasite leaves the liver cells and the red blood cells, and basically these cells then die, and this is the way that also the heme is then discarded in the end. So the drugs that are on the market, for example, chloroquine, are believed to inhibit this hemozoin formation. So the heme forms and it precipitates in the form of hemozoin. And if this can be inhibited, then, it's like when you have the waste and you can't bring it to the waste site, but it accumulates in your household. First it's in your kitchen, and all your kitchen is full of waste, and you can't prepare food anymore. And then it's in your bedroom, and you can't sleep anymore. And then at some stage, you will finally die because you don't get food and you don't get sleep anymore. And it's a bit like this. The heme here is accumulated, and it cannot be discarded as hemozoin, but it's accumulated in a food vacuole. And by the end of the day, then it's just too much. And then basically the parasite suffocates in its own waste. Yeah? So this is believed to be the action of some of these very potent antimalarial drugs. We have some structures here. Chloroquine, which is probably the most common one, is shown here. This is the structure of chloroquine, and it has been in clinical trials, as far as I know, for a very long time. It's, I think, since the 1940s or something. Quinine is sort of the traditional drug, and then chloroquine is a newer one. And it's very, very potent. The only problem is that now we use it worldwide on hundreds of millions of people, and you can imagine that the parasites are beginning to develop resistance. And there are some countries in the world, like in southern Africa, where more than 60-70% of the malaria parasites are actually resistant already against chloroquine. So we have a real problem here. This drug, this very effective drug, in some parts of the world is not effective anymore. This one here is a newer, artemisinin, is from Chinese medicine. I think the woman who discovered this in China received a Nobel Prize in medicine or chemistry, I think, medicine a few years ago. So, anyway, I'm going to tell you a story about this particular compound here, ferroquine, which is very famous. It was discovered, if you like, from actually a good friend of mine, Christophe Biot, who is in France, in Lille now. He found this. Christophe actually synthesized this compound in the group of Professor Brocard. And it's a success story in itself, and I'll go through some of the detail, you will actually see I borrowed some of the slides on this from Christoph. So I asked him and said, I'm going to do this course, would you mind giving me some of your slides? And you will see they come up in a second. So I really want to acknowledge Christoph's help here. He's really the best person to tell this story. The one thing, if you're just comparing the two structures, and this actually derived, you see this is the same core here, and then there's a side chain, there is some chirality that is removed, there's a ferrozine instead of this chain here, but otherwise it's a pretty similar kind of compound, yeah? There are some significant differences I've listed here. For example, chloroquine, this nitrogen and I think it's this nitrogen here, the amine nitrogen, they have a pKa values, and both pKa's are lowered here, in this case, in the ferroquine, as compared to the chloroquine because of the ferrozine presence. Also, this compound is more lipophilic because of the ferrozine here. Yeah, it's a large lipophilic body. Ferrozine doesn't have any chiral centers. Okay, fair enough. And it has the iron atom in oxidation state +2. And we already saw this, that this could be iron +2, iron +3. Think of the ferrocyaphene compounds, the tamoxifen analogs, where it was important that ferrozine was oxidized and going iron 2, iron 3. Potentially, the same is possible here, and we will learn how that affects the action of the drug. But first of all, let Christophe Biot tell the story of this compound here. And you can see it's only about 20 years or so old. In 1994, Christophe actually synthesized this ferroquine, it's abbreviated FQ. They actually synthesized this compound for the first time, and they took patents on this already back then. And think of the 20 years time span, so the clock is ticking for this compound, right? There were some first contacts soon after, so they actually did a pretty good job. They had the compound, they had the patent, and they found a partner, industrial partner, in order to advance development. You can see there were some activities going on, and a few years later they stopped because the company lost actually interest, and then Sanofi Aventis came, the big drug company, came on board. But you can see it's already a couple of years into the development. So a couple of years were lost, in a sense, because of having the wrong partner. Yeah? Anyway, Sanofi Aventis took up the work. They began development in 2004 already. There were the first phase 1 clinical trials in humans. It worked out to be really, really nice. They went to Southern Africa, Gabon in particular, because in Gabon there is a large number of resistant malarial strains already, and it turns out, and this is the particular thing about ferroquine, that ferroquine is active even against malaria-resistant parasites. So this is the great benefit of this compound. It's active against chloroquine-resistant bugs. So they went to Gabon to study this in the field. And you can see here, it was all going very well, and they had good success. They moved into clinical phase 2 trials relatively soon already, and then they had some clinical phase 2 trials also in combination therapy. But you can see here what happens in 2011. The company, Sanofi Aventis, actually declared, they made a decision to modify the ferroquine strategy. They discontinued exploration of the drug after very successful phase 2 trials, not due to the safety or the activity, unexpected findings. So the compound was perfectly okay. It was very active. There were no safety issues. Everything was fine, but they discontinued for economical reasons. Sanofi at the time had another drug in the pipeline, which still had a longer patent lifetime. Yeah, you see this is 2011, the patent was taken in 95, so this is very close already to expiration of the patent. So Sanofi Aventis eventually decided we're not going to make enough money from it, we'll stop it, but they specifically said the compound is very safe and it's very active. And you say, well, isn't that sad? I mean, it's something that is clearly a good compound, a good drug, and the company says for economic reason, we're out. Nobody's going to get this drug. So I had really great hopes for that, not just because, it's a very good malaria drug, because also it's a ferrozine and organometallic compound. I was really hoping they would bring this to the market, because that would be a big push also to the field as such. If you can say there is a potent organometallic drug on the market, that would really be good for the field also. Now, what happened is then there is this Medicines for Malaria activity, which I think is a sort of publicly funded initiative, and they, as Sanofi, obviously they had sort of kind of a bad conscience, so they collaborated with this public venture initiative, and they were beginning to produce fixed-dose combination therapies between ferroquine and this other compound, which is shown here, which was also very promising, because that seemed to be very promising. And this trial here should be completed in adults and children already this year. So I spoke to Christophe Biot, and he is still hopeful. He says this might be a little bit later, but they also said, they're still hopeful to bring this to the patients finally. Maybe not to the market, because now it's publicly funded. It doesn't need to earn a lot of money, but they're still hoping to bring it at least to the patients. So there's still good hope that finally we will have something on the market that has ferrozine in it. This compound actually, as you see, won a prize here from the United States Patent and Trademark Office because clearly of its impact. I mean, if you have a drug that is safe and very efficient and able to help people who have malaria with a resistant strain, and you have a new compound, that is clearly a big achievement. Okay, there are a number of things now. People think, Christoph and his team, they still think that this compound, interferes with the hemozoin formation. This is some electron microscopy where you can literally see the food vacuole here, and you can see the hemozoin crystals here that precipitate. This is the malaria pigment, and you can literally see the crystals of hemozoin precipitating. So these structures here, these octagonal structures, they are hemozoin precipitates, hemozoin crystals. The molecule interferes. There are a couple of things here. When you look at the molecule that can happen, there is an intramolecular hydrogen bond that can take place. There is the redox chemistry of the ferrozine that can take place. If you replace ferrozine iron with ruthenium, this is stopped. So there you have cases where you have no redox chemistry. If you methylate, if you methylate the nitrogen here, you can have no hydrogen bond. So there are many, many ways how you can finally all investigate the mode of action and the possible impact of, for example, redox chemistry, hydrogen bond formation, and so on and so on. And this has all been investigated by NMR spectroscopy, by crystallography, and so on and so on. It is believed that this hydrogen bond formation, it sort of forms this cyclic structure, and that seems to facilitate cellular uptake, because the compound, if it's in the cyclic structure to the outside, looks much more lipophilic than in the open state when it's unfolded. So this is clearly seemed to help membrane transport. So the hydrogen bond is important. Also important is the fact that this is, of course, largely lipophilic. So again, transmembrane transport is facilitated by the presence of the metallosine, the lipophilic metallosine. Hemozoin interaction has been explored by, by crystallography and so on, by computational work. There has been a screening for hydroxyl radicals. So this is now the ferrozine redox chemistry, iron 2, iron 3, which is producing hydroxyl radicals. There were some assays developed here, and you can see that, yes, really inside the food vacuole here, hemozoin, inside this compartment, you actually have a lot of hydrogen peroxide and OH radicals development. So in total, there must be a summarize here. This is the summary slide. And as I said, these slides are all from Christoph. I had just some slide modifications. This is a summary slide, how ferroquine enters the cells, the food vacuole of the parasite. And this is facilitated by the lipophilic organometallic residue. It's also facilitated by the hydrogen bond that can form, and the hydrogen bond is in fact stronger in the ferrozine derivative than in chloroquine because of the changes in pH. We know that ferroquine interacts strongly with the hemozoin and inhibits precipitation of the hemozoin. So this is the key step in its activity. So this is like chloroquine, but here we already get more of the compound into the cell. So in the food vacuole, we simply have more of the stuff. And so it inhibits hemozoin formation just more strongly because there is a higher concentration. And then finally, we could show that there are hydroxyl radicals, and so the redox-active ferrozine produces hydroxyl radicals, and those finally kill the parasites. And that is the two reasons why this compound is also active against resistant malaria. A, because you get just more into it, so resistance is always a question of quantity as well. If you just get more of the drug in, you might be able to kill even a resistant strain, but moreover, you have a second different mechanism of action, and these hydroxyl radicals are not present, of course, in chloroquine, so the parasite, which is resistant, has no resistance against hydroxyl radical formations. Yeah? And taken all together, this is really the mode of action, and it combines slight modifications in the pharmacokinetics and the uptake, and a totally new mode of action here. So it's the two that